So, hello everybody, welcome back. Now that you understand the reason for meiosis and you have a general outline of what's going to occur during meiosis, let's look at the individual stages. So let's get started. Each round of meiosis can be divided into a number of phases, just as we did with mitosis. But we will only use 4 phases to describe meiosis. So in meiosis 1, the 4 phases are, and this is not surprising, prophase 1, metaphase 1, anaphase 1, telophase 1. And when you see a 1 or a 2 after a phase name, you know that you're talking about meiosis and not mitosis. So let's look at prophase 1. In prophase 1, the chromosomes condense, just like in mitosis. But here's where things get interesting. The homologous chromosomes loosely pair up and they align to each other gene by gene. And this process is called synapsis. And the pair of chromosomes at this point is known as a tetrad. And I'll show you a picture of that in a second. This is very different than mitosis. Next, and this is really freaky, non-sister chromatids exchange corresponding DNA segments. And this process is called crossing over. Crossing over is unique to meiosis. Each pair of chromosomes usually has 1 or more regions where crossing over occurred. And so let's take a look at it on the next slide. Here is prophase 1, and you can see that the chromosomes are lining up next to each other, gene by gene, and then they're exchanging parts. It's pretty cool, actually. So as a result, the chromosomes are now going to be a mixture of this organism's mother's and father's chromosomes. They will no longer be either the mother's or the father's, but a mixture. And you can see that if we go over here and look at Anaphase 1, where they're being pulled apart. You can see this one has a bit of one parent and a bit of the other in this part of the chromatid, and the opposite is true up here. So that is the process of crossing over. When they're lying next to each other like this is called a tetrad. Next, we'll go on to metaphase 1. Can you guess what happens here? Well, it's different from mitosis, but similar. In metaphase 1, the homologous chromosomes line up at the metaphase plate with one of the homologs facing each poles. And this, as I said, is different than mitosis. The microtubules from the spindle are attached to the centromere of each chromosome. And then you know what this is in preparation for, pulling them apart. But let's look at, here's the diagram of metaphase. You can see that the chromosomes are lined up along the metaphase plate. In anaphase 1, guess what happens? Right, the homologous chromosomes are separated. One chromosome moves towards each pole, guided on the spindle apparatus. The sister chromatids remain attached at the centromere, and therefore they are replicated chromosomes that are moving apart. And the result is that the daughter cells are going to be haploid. Let's take a look at anaphase 1. Here you can see that the homologous chromosomes, which are all replicated, are being pulled apart. The next phase, telophase and cytokinesis, plays out differently in different species. But we'll try to give you some general rules. In some species, the nuclear membrane begins to reform. In others, the cells immediately go into prophase 2 as soon as cytokinesis happens. Cytokinesis occurs simultaneously. In animal cells, a cleavage furrow forms, just like we showed in mitosis. In plant cells, a cell plate forms, just as we looked at earlier. But there is no interphase, and there is no replication between meiosis 1 and 2. And the reason for that is the chromosomes are already replicated. Here is telophase and cytokinesis. And you're also seeing meiosis 2 is going to look like. So you can see here the nuclear membrane is starting to reform. The spindle apparatuses are dividing and getting ready to move to the poles. So let's go on. In meiosis 2, one thing you should realize is that it is almost identical to mitosis, but it begins with a haploid cell that has replicated chromosomes, not a diploid cell that has replicated chromosomes. In prophase 2, a spindle apparatus forms and moves towards the pole. In late prophase 2, chromosomes move towards the metaphase plate. There is no crossing over that's going to occur, no synapsis. So you can see that the chromosomes are starting to move towards the center of the cell. They're starting to recondense if they've decondensed a little bit. Can you guess what happens in metaphase 2? Of course you can. In metaphase 2, all the chromosomes that remain align at the metaphase plate, the equatorial plane, and because of crossing over that occurred in meiosis I, the two sister chromatids are no longer genetically identical. And then the microtubules attach to the centromeres of the sister chromatids. So let's take a look at that. Here are the replicated chromosomes lined up on the metaphase plate, and it's showing you what's happening in each of the two cells that were produced by meiosis I. In anaphase 2, I bet you can guess what happens. Yep, the sister chromatids separate towards the opposite poles. You can see it looks just like mitosis on a haploid cell. And then finally, in telophase 2 and cytokinesis, just as you'd expect, the chromosomes arrive at the opposite poles. The chromosomes begin to decondense and the nuclear membrane reforms. When the nuclear membrane is complete, meiosis is finished, and then cytokinesis separates the cytoplasm. At the end of meiosis, as a result, there are 4 daughter cells, each with a haploid set of unreplicated chromosomes. Each daughter cell, therefore, is genetically distinct from the others and also from the parent cell. So here you can see telophase 2 and cytokinesis. You can pause the video if you want to look at that longer. So now that we've studied all that, we want to compare mitosis and meiosis just to make sure that it is clear in your mind. Mitosis conserves the number of chromosome sets. In other words, in mitosis, the daughter cells are genetically identical to the parent cells. Meiosis reduces the number of chromosets from 2 to 1, from diploid to haploid. And it also produces cells that differ genetically from each other and from the parent cell. And they differ from the parent cell in that the chromosomes have been recombined during crossing over. These 3 events are unique to meiosis, and all of them occur in meiosis I. First is crossing over in prophase 1. The second is the alignment of homologous chromosomes, the tetrads, at the metaphase plate in metaphase 1. And the third is the separation of homologous chromosomes at anaphase 1. So those are all different. You should make sure those come to mind readily. Here is a diagram of what I just said. Here we have mitosis, and you can see that the haploid number here is 3 and the diploid number here is 6. And here we have 6 replicated chromosomes, and in the daughter cells we have 6 unreplicated chromosomes. Here we have the same haploid number of 3 and diploid number of 6. And you can see here that we begin with 6 replicated chromosomes. In meiosis I, we create haploid cells. They each have 3 replicated chromosomes. And that after meiosis II, we have 4 cells that are all haploid. They have a chromosome number of 3, and the chromosomes are unreplicated. This might be a very worthwhile drawing for you to hand draw and put into your notes. I have a feeling that if you did that, you would not need to do any further studying. Here's a table summarizing what happens in mitosis and meiosis. Maybe this is useful for you. I'll let you look at this on your own time. In the next talk, we'll be going on to look at some topics that have been raised by meiosis that we didn't completely cover today. And so it'll be a bit of a smorgasbord where we'll talk about a lot of odds and ends. Until next time, go outside, take a look at nature, ask biological questions, bring them back to class, and I'll see you real soon.